Hello guys, how are you all? I hope you are all doing well. So welcome to my YouTube channel. It's on educational planet. So our today's topic is post-fertilization events. So let's get into the video. Post-fertilization events. Endosperm development. So what is endosperm development? As I have already told you in my last video that how does the endosperm develops when the syngamy and triple fusion forms, which form this syngamy and triple fusion forms the peg, which is primary endosperm cell. So when the syngamy and triple fusion forms peg, which is primary endosperm cells, which later develops into endosperm. So how does the endosperm develops? This I have all told you in my last video. So now let's learn more about how does the endosperm develops. So first, endosperm develops first, followed by an embryo. Endosperm develops from pen, which stands for primary endosperm nucleus. Primary endosperm nucleus. फर्स्ट ऑफ ऑल जो एंडोस्पर्म डेवलप होता है वो एक एम्ब्रियो की वजह से होता है, एंड फ्रॉम वेयर प्राइमरी एंडोस्पर्म न्यूक्लियस द पेन अंडरगोज सक्सेसिव nuclear divisions to give rise to free nuclei, and this stage of endosperm development is called free nuclear endosperm. Means when pen undergoes nuclear divisions which give rise to free nuclei, and this stage of endosperm development is known as free nuclear endosperm. Subsequently cell wall is formed. So सेल वॉल फॉर्म हो जाती है एंड एंडोस्पर्म बिकम्स सेलुलर. तो एंडोस्पर्म बिकम्स सेलुलर. Then the next point is this division is followed by cytokinesis, and thus endosperm becomes cellular. It is now called cellular endosperm. Then this division follows by cytokinesis, and thus that's why also endosperm becomes cellular, and it is now, you can call it as cellular endosperm. Then what are its functions? The cells of endosperm tissue are triploid and filled with reserved food materials to nourish the developing embryo. So 3-marker question can come also. What are the functions of endosperm development? So you have to learn them. Second point is the water of tender coconut in the center is free nuclear endosperm, and white kernel in the outer part is the multicellular endosperm. Then the next point is the endosperm may be completely consumed by the developing embryo, for example pea, beans, or it may persist in mature seed, example coconut. Then our next is embryo development. Embryo develops from zygote at the micropylar end of embryo sac. So when does the embryo develop? At the from zygote, first of all from zygote, and where at the micropylar end of embryo sac. Then the second point comes is the nutrition for development is provided by the endosperm. So how does the nutrition provided by the endosperm? Then next point is the zygote divides to form proembryo. Then the next point is the different stages of the developing proembryo are globular and heart-shaped embryo, which finally forms mature embryo. Then our next topic is embryo in dicots. The zygote undergoes transverse division, forming a large basal cell and a small apical or terminal cell. Then next point is the large basal cell enlarges and undergoes transverse division to form a group of 6 to 10 cells called suspensor. Next point is the first cell of the suspensor towards the micropylar end is called hosterium, whereas the last cell of the suspensor towards the chalazal end is called hypophysis, that later develops into radicle. So the first cell which is of the suspensor towards the micropylar end is known as hosterium, whereas the last cell of the suspensor towards the chalazal end. So you have to learn it, you have to remember it, that the first cell of the suspensor towards the micropylar end is known as hosterium, whereas the last cell of the suspensor towards the chalazal end is known as hypophysis and later develops into radicle. Then next point is the smaller terminal or apical cell undergoes one vertical division. The two cells formed from terminal cell divide by a transverse division, thus forming 4 embryonal cells. Quadrant stage. When the 4 embryonal cell forms due to a transverse division is known as quadrant stage. Quadrant means 4. So 4 embryonal cell is also known as quadrant stage. Then coming up to the next point, finally these 4 cells divide vertically. Then these 4 cells vertically divides, forming 8-celled proembryo, that is known as octet stage. So what is octet stage? When the 8-cell proembryo have been divided by the 4 cells vertically is known as octet stage. And what do you mean by quadrant stage? When the 4 embryonal cells were divided by transverse division of the 2 cells when they are formed are known as quadrant stage. Means जो 2 cells होते हैं वो form हुए होते हैं वो ट्रांसवर्स डिवीजन से फॉर्म करते हैं 4 एम्ब्रियोनल सेल नोन एज क्वाड्रेंट, वेयरएज जो ये वाले 4 सेल्स होते हैं ये फर्दर डिवाइड करते हैं वर्टिकली, जो कि 8 सेल प्रो एम्ब्रियो बना देती हैं, विच इज़ नोन एज ऑक्टेट स्टेज. 4 cells at the apex give rise to plumule and another 4 give rise to hypocotyle except its tip. मीन्स जो 4 सेल्स होते हैं एपिक्स पे वो गिव राइज करते हैं टू प्लूमले, एंड जो अदर लेफ्ट आउट 4 हैं वो हाइपोकोटाइल पे लेते हैं एक्सेप्ट इट्स टिप. टिप को छोड़ के बाकी वो होते हैं. देन नेक्स्ट इज स्ट्रक्चर ऑफ dicotyledonous embryo. A typical dicotyledonous embryo consists of an embryonal axis and 2 cotyledons. The portions of embryonal axis above the level of cotyledon is called epicotyle, which develops into plumelet or baby shoot, known as also future shoot. Third point, the cylindrical portion below the level of cotyledon is the hypocotyle, which develops into radicle or root tip, known as future root. So in this you have to draw the diagrams, you have to learn the diagrams, then you have to explain it, which will also help you to secure your good marks. Coming up to the next point, number 4, the root tip is often covered with a root cap, calyptra. Mango, apple, radish, rose, etc. are some dicot plants. So a 1 marker question can also come. Name some dicot plants. So mango, apple, radish, rose, these are the examples of dicot plants. Coming up to the last topic, which is the structure of monocotyledonous embryo. First point, a typical monocotyledonous embryo consists of one cotyledon. Only it consists of one cotyledon. As we can understand by the name, mono means one cotyledon. Second point, the cotyledon is situated on one side, that is lateral, of the embryonal axis is called scutatum. Third point, the radicle and root cap are situated at the lower end of embryonal axis in an undifferentiated sheath called coleoryza. The shoot apex and few leaf primordia are enclosed in a hollow foliar structure in epicotal region called ciliopatile. Then next point is the portion of the embryonal axis above the level of attachment of scutellum is called epicotyle. Then last, what are the examples? 1 marker also can come. What are the examples of monocotyledonous embryo? That are monocot plants, that are grass, banana, bamboo, palm, etc. are the examples of monocot plants. So guys, thanks for watching my today's video. So we will meet in the next video with another topic. So guys, please like, subscribe, comment down below and press bell icon for more notifications. Thank you so much guys. Bye bye.